Today, let's focus on how to exploit the genetic variance using genomic prediction. I would like to maybe start just summarizing some of the learnings from yesterday and the day before. Yesterday we were talking about on optimizing selection intensity and cycle time. Then the day before on maximizing accuracy, since we propose this step-by-step approach. Today we will discuss a little bit about the use of genomic prediction to exploit genetic variance, some of the challenges when it comes to logistics. But I would like to take advantage of looking at this roadmap, like how do we propose that you could implement genomic prediction first to basically come up with best or better surrogates of genetic or breeding value in your program, using it in the field trials, where we were talking about how the accuracy can be increased drastically when the heritability is low, because basically your training population and the individuals that you're predicting are the same. So you're observing the individuals and you're observing the haplotype, so accuracy is high. And then moving to a second step on optimizing the selection intensity, where now you predict individuals that you have not observed, but because you are predicting full SIBs or related material, you will actually have observed the haplotypes in the case that that's what you're trying to predict. And then reducing the generation interval or cycle time. We talk about that this is a bit more difficult because basically what you're trying to predict, you have not observed neither the individual, neither the haplotype. So that becomes a bit more difficult. And Joseph was showing a practical example of what type of accuracies you can expect from these different approaches. If you go for number one, you will find pretty good accuracies. If you go to number two, you will have a bit lower accuracies, but it's still pretty high. And you go to number three, trying to predict something that you have not observed, either individuals or haplotypes, then you expect lower accuracies, but to be compensated by cycle time reduction. We have also discussed that all this comes as further improvements after you have adopted maybe selection indices, good experimental designs, etc., etc. And all this will help you in two aspects: improving genetic gains, but also delivering better products, faster release of products, more accurate selection of products, testing more products. So you can see the two sides of the coin: genetic gain, but also product development improvement. And when we talk about genomic prediction, to exploit genetic variance, basically we are talking about doing better crossing strategies. There is two things. In the case of population improvement, you want to guarantee that the allele frequencies and the haplotypes are inherited to move the population means for the additive, and in the case of non-inbred individual crops, also for the non-additive components. Whereas in the product development, is more about identifying missing opportunities, like hybrids that you have not produced, or some kind of missing opportunities. So I'm putting the crossing strategy, normally at the beginning of the pipeline, but in the case of the hybrid programs, there might be additional crossing components. But in general, everywhere where you are making some crosses, basically that's a room for exploiting the genetic variance. So let's jump into the topic to don't further delay this. And I want to start by basically going back to the example that I shared with you yesterday when we were talking about applying genomic selection to reduce the cycle time. As you may remember, we were first comparing some of the classical strategies and then the introduction of genomic selection, and we identify a big issue. Basically, when you pull the trigger to reduce the cycle time, drastically, thanks to the prediction methodology of decoupling the phenotyping from the selection of parents, what happens, if you may remember, there was a big change in genetic gain, but also reaching a plateau. And what happens really on the background is that, when you have a conventional program, let me start this figure little by little. This is just, an idealized representation of how a breeding program works in terms of cohorts moving across years. So if you were to create a breeding program from scratch, and you would take, let's say, the best 20 individuals in the first year, and you make some crosses. This, of course, is just focused on the line program, but the idea extrapolates to also non-inbred individuals or non-inbred crops. You would start, let's say, in year one, making some crosses. Maybe if you have a good system through SSD, you might be able to run more than one generation per year. And in general, the point is that when you finish the first year and you go to the next year, you are still not able in a conventional program to recycle parents because you have not generated enough information to ensure that you are recycling materials with good breeding or genetic value. So most programs still the following year will, let me put my pointer, the following year will make some new crosses and so on. So what happens in regular programs is that you have actually multiple cohorts moving across in your program. So even though in the crossing block, you may have a certain effective population size, the effective population size of the program is actually much higher, depending on how many cohorts you actually have in your program. And actually you have as many cohorts as the time that it takes you to reach the first decision or the recycling decision. So if it takes you, let's say, about 5 years to reach, let's say, stage 2, when you consider that you have enough information to recycle parents, that means that probably if you need 5 years, then you are going to have 5 cohorts moving in your program. So it would be like multiplying the effective population size of each crossing block by the number of cohorts. Now, when you actually enable genomic prediction, what you do is that you decouple the product development from the recycle of parents. Decouple the phenotyping from the selection of parents. So that means that you can actually start selecting parents immediately. As soon as you produce some individuals, you predict them and you recycle them. That actually reduces drastically the effective population size of your program. And you know, that acceleration really leads to some drift and less genetic variance in your program in general. So yesterday, I was showing you that the immediate outcome is that the genetic gain that is translated, that uses the genetic variance to basically translate it in genetic gain. It moves so fast that you reach a plateau very quickly. So this is what I would call, let's say, the variance burning rate. Think on the genetic variance as some sort of fuel or gas. This acceleration basically is burning the fuel in not only much faster, but kind of inefficiently, because the translation of genetic variance in gain gets reduced due to the drift. So the use of genomic prediction for genetic variance is about controlling that burning rate. And yesterday, basically, I presented to you 3 ways to control that issue. Again, just imagine the conventional program is moving, let's say, slow, but it's ensuring that you use that gas or that fuel in an efficient way. Whereas in the case of going super fast with genomic selection, is like, poof, you press the gas pedal and then you start to go super fast, but you are burning that variance in an inefficient way if you do not take additional considerations. Yesterday we were talking about, I know that you cannot see the little labels, but the first figure is showing in the y-axis the genetic gain, as we saw before in other plots. In the x-axis, we see the time in terms of years of the breeding program that is running. And then the 3 lines represent the conventional program in the most aggressive possible way. And the red line represents the typical genomic selection, where you just reduce the cycle time as much as possible. And then the green lines, the green line represents, in this case, the solution to basically lower the burning rate. That is optimal contribution. In the case of optimal contribution, you put some weight to the genetic variance and some weight to the performance. So somehow you're saying, let's sacrifice some of this performance to ensure that we can actually, lower the burning rate and kind of keep the genetic gains for longer time. So you're sacrificing a little bit of performance, but actually you will gain more performance in the longer term. Then the other approach to basically control for this accelerated burning rate, more than accelerated, this inefficient burning rate, is to separate the burning of the simple and complex traits. As we mentioned, that in the case of the, what we noticed is that in the case of the simpler traits, the burning rate was much faster, and the complex traits was going okay. But by separating the selection for complex and simple traits, you can again achieve a better result. And also yesterday we said, okay, another possible solution is if I'm basically burning the gas too quickly, let's put more fuel into the machinery. And that was the third option that we were showing. Okay, what if you increase the effective population size? So instead of having a crossing block of, let's say, 40 individuals, what if we have 80, 120, 160, 200? And there's a limit. It's like saying, okay, this machine can only take that much gas. Let's just have certain number of individuals. I think I also showed this figure that you can see that some of the methods really achieve the purpose for different traits. We were showing that in the case of simple traits, which were trait 1, trait 2, and trait 3, and the complex traits, the next 5 traits, some of the methods basically were addressing the issue of drift, whereas in some cases it was not that successful. In many of them it was. So an important note is that the accelerated burning rate of genetic variants is only an issue in genomic selection programs. This is not an issue in conventional programs. So I know that that you are also going in that direction, but try to don't lie to yourself, telling you that you are consuming all the genetic variants if you have a conventional program. I mean, if you knew, I mean, how slow the allele frequencies for complex traits really change, actually you wouldn't mind, or you wouldn't be concerned of using, for example, 20 parents in your crossing block. I mean, you could do breeding for 100 years if you have a conventional program that has those small crossing blocks. Of course, always keeping in mind that you have to bring some genetic variation for new traits that the market might be requiring. So conventional versus genomic selection programs. As we mentioned, just a Mickey Mouse example, maybe the effective population size in your program is about 50 because you have multiple cohorts. Really, that can last for more than 100 years for many of the traits, because like I said, actually allele frequencies really don't change that fast when you're talking about complex traits where you have maybe hundreds of QTLs in that trait, or some may speculate that even thousands. So the considerations in that case, in the conventional programs, is to, okay, you still have to take, exploit the genetic variance properly. So, but that can be achieved by balancing the selection of among and within family to ensure that you are exploiting both sources of variation. And when it comes to crossing the materials, well, just make sure that you try to couple materials that are non-related, and that you're also selecting, like I said, some individuals in each of the best families. And in the case of the GS programs, is really when you have to think how to use that genetic variance more carefully and properly. It's not only the same that, you need to still select among and within family, cross non-related individuals. In this case, you can actually come to better approaches, like, predict crosses with high performance and high variance. I was mentioning optimal contribution. You could also use something like genomic prediction of cross performance. We might get into that later. Some other people call this also breeding starts, where you can predict the progenies and make sure that you are focusing on bringing not only high performance families, but also families or crosses that present high genetic variance. Like we mentioned, there might be other solutions, like adding more genetic variance to the crossing block when the burning rate is too fast. and like I said, considering the simple and complex trait architecture into your selection, because like I said, the effective population size will really be reduced when you move to a genomic selection program. So this is just a figure that I thought from some of the Roslin people. You know, sometimes you have to just kind of leverage from genetic gain and diversity, and you have to come to a trade-off. And sometimes, of course, that trade-off, most of the people in some of the CG programs have gone in this direction, trying to keep a lot of diversity, but sometimes with very low genetic gains. I think, of course, you need to find a good sweet spot for your purposes, but normally, with the levels of variation and germoplasm that exist in the programs, sometimes it's good to, go for a genetic gain and of course, keeping in mind the diversity, but coming with a clear strategy, not just about bringing as much materials as you can find. Okay, so some of the learnings, an aggressive and an accurate GS strategy will exploit the genetic variation faster than phenotypic selection. That is good because that's what we want to do. But again, this will bring faster genetic drift that will basically hurt the program because you're gonna probably lose some useful alleles. And we are providing to use some recommendations on how to deal with that burning rate of the genetic variance. And the genomic prediction approaches can be, optimal contribution, but it can be also, using things like genomic prediction of the crosses that can occur and that will bring more genetic variation in the next generation. Let's move to some of the technical part, like really, okay, what marker density do I use? What type of training population? What genotyping, etc. This, let's start, okay, with how many markers do we need? To investigate how many markers do we need to implement genomic selection, and I'm going to focus specifically for the reduction of cycle time. And remember, this refers to predicting haplotypes and individuals that we have not observed. So we investigated different number of markers, and we kind of fix the other variables like number of individuals and composition of the training population to certain values and assume that there is not going to be a strong interaction between the different variables. So we could fix some factors and vary the others. In this case, the one that we are varying is the number of markers. So let's, as we were talking about the genomic selection, we're talking about building the training population from the stage 1, stage 2 materials produced at F5 and F6 in the BEANS program, then predicting the F1s and using those individuals to select parents. We were using also OCS or optimal contribution, basically putting some weight to genetic variance to ensure longer term genetic gains. And then what we were varying is the number of markers, 500, 1,000, 2,500, 5,000, 10,000, and 30,000. And let's go to the results. What we found is that, the number of markers, maybe let's focus instead of a trait by trait, let's focus first on the selection index that represents all the traits. What we found is that the number of markers had only an effect after 10 years of running the accelerated GES system. It's really until that point that you can start to see differences of having lower versus higher densities in the genomic prediction approach. So for example, when we were using 500 markers, you started to notice that, okay, genetic gains were reaching a plateau. Then also we were with the 1,000, it was then reaching a plateau after this number of years. But actually having many more markers, at least for this time period, it was not making a difference. Of course, if we extend this for more years, maybe 30, 40, 50 years, probably you would start to see that also they start to separate the 2,500 from the 5,000, et cetera, et cetera. But at least for the purpose of this program, we were looking at a 10-year time window. And that seems to be, probably, about more than 2,500 markers it was more than enough. So 2,500, but maybe 5K would be something between 2,500 and 5K. That was probably more than enough. And when we look at a closer reason why this is happening, instead of looking at the genetic gain and per trait or across the index, we could look also at the accuracy that we are reaching at the F2s. And basically, we could see that for certain traits, for the lower density markers, the lower density panels, let's say 500 and 1,000, we basically were breaking the accuracy in the model. You know, this is expected because we know that, you know, that we're going to start seeing more haplotypes. You know, they start to break, and basically the density that you have is not able to capture those haplotypes by the linkage disequilibrium. And that's okay. I mean, that's kind of expected, but I want to emphasize something here. Let me actually skip this for a minute and just come back to this slide that we were talking about. I'm referring specifically to the case where you want to reduce the cycle time, and basically you're trying to predict something that you have not observed. That's where you need, you know, markers in the order of 3,000, 4,000, 5,000, etc. When it comes to really using genomic selection for the purpose of number 1 and number 2, that basically you just predict the individuals that you already observed, or predicting individuals that you have not observed, but you have observed the haplotypes because you have very closely related individuals in sparse testing, you actually don't need that many markers. You can actually go to the literature and look at the classical papers from Lawrence and Bernardo, etc. You only need the markers in the order of hundreds to do a good genomic prediction in these two scenarios. It's only when you move to the aggressive component of GS that you need to put markers in the order of thousands. So I will not repeat what the Eng and the team from Module 3 was presenting, but you know, some of the common genotyping systems are the SNP arrays, GBS, whole genome sequences, but really, like I said, the linkage disequilibrium is high. In the case of the number 1 and number 2, linkage disequilibrium, it starts to, you know, be more difficult to capture when you move to reduction of cycle times. You need more markers, but nothing that you should worry that much. Let me move to the next question that probably you are interested. Which model to use? Okay, so when it comes to what genomic prediction model to use, I will not elaborate much because I think that's where Abhishek will provide a good insight. I guess the only thing that I can say is that there has been great progress to understand the trait architecture and how can this be taken into account by different modeling approaches, you know, the Bayesian alphabet and now the, you know, the machine learning approaches or support vector machines, etc. But, you know, at least up now, despite you know, the many options that you may find, this is not the newest paper, but you know, there's many options, and this keeps growing. Now there is more and more approaches. All of them look actually or seem to work pretty good. I don't want to make any enemies in the session, but I think a more complex model will not be actually the game changer for your GS implementation. You know, the current REML frequentist approach keeps being the workhorse in many institutions, companies, programs, where you can actually model very complex G/E stuff, genomic relationships, etc. And of course, some of the newest methods that are in the literature might be good to account for different, like I said, trait architectures. But I would really advise that you don't focus too much on this part until you have figured out basically the basics, having databases, having analytics, etc. Later on, you can focus on some of these things. Let's go very quick to the training population. You know, the general advice is that when it comes to defining what is going to be your training population, as I mentioned at the beginning, try to use, and this is really probably the most important recommendation for training population, use as training population, the material that is generated, the phenotypes that are generated in your program. Everything that your product development pipeline, stage 1, stage 2, stage 3 is producing, put it in the training. So phenotype those individuals, genotype those individuals. So after a couple of years, maybe 3-4 years, you have enough data there to start, running the machine. Make sure that you have a closed system. As we mentioned, the genomic prediction models really work based on the relationship that exists between a training set and a predicted set. So make sure that that relationship exists by basically closing the system and avoiding that foreign material gets in the crossing block in big numbers or too often. And if you're going to introduce some material, be very careful on how you do it. Still, some special traits might be the special training populations. It might be that your product development pipeline is not producing the phenotypes for certain traits. Then you might have to produce a special training population, but this will not be the rule. It might be just the exception. So, watch out for that. And of course, also something important is that you need to keep updating that training population. How big? In general, just if you have more genotypes and more phenotypes, put it into the training model. Put it into the training model. Is very unlikely that if you have a closed system, you will have any damage by putting more information there. Many of the papers that say that you can lose some prediction accuracy, it is normally because some of the systems that they present are not really closed. Still, there is something that I will comment on the composition of the training population. Before we were looking at the number of markers, and now I'm going to actually fix in the simulations that we did for this program, we fixed the number of markers being used to 3,000. Still the same, the type of strategy, genomic selection using OCS, selecting in the nursery the parents. But now what we wanted to change, it was not the number of markers, it was not the number of individuals. It was just, it was 5,000 markers, 3,000 individuals. But what we wanted to change, it was whether should we use, all data? Should we use the most recent data? Or should we use random data? Kind of, if we have a database with thousands and thousands of records, that these models actually are, quite computationally intensive. So if you just say, okay, I'm going to bring 100,000 individuals just because I have them in the database, that also might not be a smart approach. So something that you could do is to, okay, let's think on what a normal GBLOB model can take. And, probably some of you that have fitted some of these models, that really already putting 10,000 or 20,000 individuals can affect drastically. But first, before looking at how many individuals to put in the model, we wanted to see if there was an effect between putting all data, newest data, or just a mix of all the data, so old and new. And actually, what we found is that as we expected, if you the older data, which we were calling here first, or first records, after a certain number of years, in this case about 13 years, the model started to kind of get, it was not being updated somehow. So that was one of the recommendations that we were saying, try to update the model. This would basically represent the scenario where you do not update the model, so the accuracy starts to go down, and as a consequence also the genetic gain. So there's a limit of how useful is the information in certain time period. You need to keep updating your program. And actually the best, of course, was the one that was actually using the newest data. If we were going to use always 3,000 records to fit the model, it's better to use the newest records because that keeps the model updated. And something that was mixing between all the new records, of course, got something in the middle in terms of genetic gain for the index, but also for some of the traits, which was behind the index. And of course, like I said, that is because the accuracy, look at the one that is using all records, basically the accuracy of the model is getting lower at a faster rate because it's not getting updated. And that happens for several traits. So that's what is happening behind. The model is losing the accuracy because it's not being updated. So let me move to the last question, and then we can take your comments and try to answer any possible doubt. Something that we also wanted to explore was how many individuals we should have in the training population. Again, because of the time that it takes to fit these models, and when we run these simulations, we are simulating entire breeding programs, sometimes for many replications. So maybe sometimes one of these replications, it takes a week to run, because you are basically simulating that you are running a breeding program for, let's say, 20 years, and that every year is feeding one of these models, and that is feeding many other models, for the regular breeding program. So that actually takes time. So we didn't explore many individuals in the training set, but at least we explore some of the small numbers to something intermediate. For some of you know that maybe a GBLOB model with 5,000 individuals, it might take up to 20 minutes to run just for a single trait. And remember, we are running it for multiple traits, for multiple years, for multiple replications. This is what we vary. We were fixing the rest of the factors to attribute the differences to the number of individuals or records that we put into the model. So we already knew, okay, these many markers, more than 2,500 should be more than enough for 20 years. We know that we should use the newest records, so keep updating the model. And now we wanted to check what happens if you put more and more data into the model. Here you can see the results for the index and also for the specific traits. Again, in the y-axis, you can see the genetic gain, and in the x-axis, you can see the time that the program is running. And you can see that after 5 years, the size in terms of the model and the number of records that we use, it starts to have an effect. All of them are using the same approach in terms of sampling the records. But the difference is that, that basically when you have less records in your training model, the accuracy of the model is lower. So you can see that the genetic gain is lower when you were using only 500 records. If we were putting always 1,000 records every year, so every year that we were feeding the model, we were taking the newest 500, the newest 1,000 records, the newest 3,000 records, the newest 5,000 records. And you could see that, you start to get higher and higher. Probably we were feeding 10,000, which was kind of too much, because that would take probably a couple of hours to run just for a single trait, single iteration. It would probably not go much farther away. So again, this is because these are closed systems. If you go to the animal breeders, because they do not have, necessarily many individuals per cross, they don't have those strong relationships or much data building on those relationships. They normally require thousands and even millions of records to have good accuracies in their prediction models. But in closed systems that use recurrent selection, that control very well the flow of germplasm, actually you can expect that you can just sample several thousands of records. In this case, the best was 5K. I assume that 10K will still be a bit higher. But let's say you just take as many records as you have till the point that you can still feed a model in a practical time. You might have 100,000 records from the last, I don't know, 100 years, but it might be more practical to just take the newest records, maybe the newest 10,000 or 15,000 records, where you can still feed a model for multiple traits in considerable time. In many breeding programs, the turnaround times need to be very fast. If you only have a week to wrap all those models, provide all the predictions for all the traits, etc., you may not want to feed more than 15 or 20,000 records in some of these models. But again, of course, that will depend on the structure of the breeding program. And again, looking instead of the genetic gain, looking at the accuracies. I'm not looking too much at the variances because it's not too much of an issue here. But when you look at the accuracies, you can see that this is the reason. I mean, when you have less individuals, basically the accuracy of the model is lower. And that is what is driving genetic gains to be lower with less individuals. It's not the fact that there is just less individuals, it's the fact that the accuracy is lower. And that's everything that I wanted to present. I'm just finalizing with what we've been talking about all these days. What is the path to implement genomic selection step by step? Looking at something simple first, trying to get some genotypes for your field trials, your stage 1, stage 2, and then once you have adopted something like sparse testing, moving to implement genomic prediction as an additional layer, and then moving to the, because at this point, if you are able to achieve this, is because you already have some good analytics in place, you have some good experimental designs, etc. Then you can start thinking on implementing genomic prediction for reducing cycle time in a recurrent manner. And then by doing this part, you're going to have to immediately tackle how to exploit the genetic variance better through genomic prediction approaches. And maybe something that I didn't mention too much because there was no time yesterday is that we need to actually start thinking on the selection indices, because this will be instrumental for enabling genomic prediction when it comes to reducing cycle time. Some of the programs, I'm going to cite the IITA Cassava program, they did a great effort to implement this part with a lot of help from the Cornell University, the donors, etc. But what they notice is what we see, that the genetic gains, boom, exploded. They went super high for yield, but then when they started to look at other traits, they noticed that they were going in the opposite direction, or they were going in a direction that they didn't want to. And that's because, you should never talk about doing genomic prediction only for one trait. You should do genomic prediction to basically obtain phenotypes that you couldn't obtain in any other way by getting those predictions. But the selection is still the same in the sense that you need to look at multiple traits. You need to really apply selection in this complex world of finding trade-offs between traits. When you are in a conventional approach, that maybe some of the traits you're observing in the field and you go and you select them visually and some others in one stage or some others in another, that's maybe more intuitive of what you do currently. But when it comes to use genomic prediction, you have to put all those selection strategies in a single time point, let's say in the nurseries or the early stages. You need to know how to use all those phenotypes, combine them, select parents that will ensure that you still make the gains. It's just that now you are not observing those individuals.